There's a hidden world inside each of us. Ever since technology revealed the nanoscale constructs of biology, we humans have tried to take the machine apart. But more often than not, when we reach for the ultra-fragile nanoverse, it slips through our fingers. Not Steve Block's. In his Stanford lab, he can deftly manipulate the tiniest components of life down to a single molecule, suspended in calipers of light.
I was the son of a particle physicist, and it's fair to say that I've lived most of the places in the world where there's a particle accelerator.
Particle physics largely happened in Europe, far from his home state of North Carolina. Life abroad often left Block homesick.
When I was a student at Oxford, I wrote off to my sister. I said, "Send me some American music before I go crazy." She sent me 2 cassettes. One was Earl Scruggs' famous Foggy Mountain Banjo album. My jaw dropped. I couldn't believe the music I was hearing.
Block immediately bought a banjo.
It was either that or a set of bagpipes, but my fellow students told me if I came back with bagpipes, they would wrap them around my neck and throw me in the college lake.
Block mastered the banjo and took to the stage while earning his PhD in biophysics from Caltech. Touring America's sprawling bluegrass circuit, his thoughts increasingly became occupied with motors. Very different motors from the one in the Volkswagen bus, carrying him from stage to stage. These were the smallest engines in biology. Molecular machines like those that cause muscles to contract, cause DNA to unzip, motors that make life possible. Block wanted to know, were their motions fluid and continuous, or incremental, coming in discrete steps like notes up the frets of a banjo? One stubborn fact of physics stood in his way.
To study a molecular motor, Block needed to pick one up and watch it turn. You might imagine something like chopsticks picking up a grain of rice. But at molecular scales, our everyday concept of tweezers begins to fall apart. You can't reliably grab hold of a single molecule using other molecules without destroying, chemically changing, or even losing the first molecule.
A paper from 1987 sparked a light bulb moment for Block. A device invented by physicist Arthur Ashkin that could trap molecules with light. Block got to work building one of his own.
Believe it or not, at the microscale, a focused beam of light creates pressure that can suspend a single particle in the air. Block realized that such a particle, just a tiny bead, could be the perfect anchor for the even tinier molecules he wanted to study. By chemically attaching a biomolecule to the bead, he could then measure the minuscule forces of its movement.
The apparatus is called an optical trap, or optical tweezers, and with it, Block began to revolutionize biophysics. In a landmark experiment, Block precisely targeted the invisible corkscrew-like tail rotor by which bacteria propel themselves. Tail frozen in the optical vise, the whole bacterium spins like a propeller. Amusing as they are to watch, experiments like this had profound implications. Scientists finally had a way to see and test processes that could only be studied mathematically. Block's groundbreaking experiments unlocked the mysterious world inside human cells, demonstrating the hypnotic mechanics of DNA processes, how proteins move and fold, processes critical to disease and medicine. With a system of lasers, Block founded an entire scientific branch called single-molecule biophysics, laying the foundation for something much bigger, a comprehensive understanding of biological life.
Like many other scientists, I believe that there exists an objective universe. There exists a reality. You don't learn about it by simply taking accepted wisdom. You have to go in there and look at it. You have to learn truth by experiment, by testing. There's so many things we don't understand, and yet the best chance we ever have of understanding of that comes through science.